Iris pigmentation.
From the Department of Pathology at the Wills Eye Hospital and the Sidney Kimmel Medical College at Thomas Jefferson University in Philadelphia, Pennsylvania.
The iris is named after the Greek goddess of the rainbow, who served as the messenger of the gods. The term rainbow membrane occasionally was applied to this structure in the older literature. This term reflects the spectrum of iris colors found in different people that range from gray-blue, hazel, brown, and deep brown that may appear almost black. Radially oriented, undulating white lines dominate the biomicroscopic aspect of the blue iris. The white lines are the collagen-clad iris vessels located in the mid or deep stroma. These are visible in lightly pigmented irides because the melanocytes in the anterior iris lack pigment and are transparent. The iris pigment is concentrated in the anterior border layer of the iris. In darker irides, large amounts of pigment in the melanocytes opacifies the anterior border layer and obscures the iris vessels and other structures in the deeper stroma. Parts of a dark brown and a blue iris are compared in these photos, showing obscuration of the vessels in the brown eye. The melanocytes in the anterior border layer of the dark brown iris are shown in this transmission electron micrograph. The cytoplasm of the melanocytes contains numerous granules of melanin called melanosomes. In contrast, the melanocytes in this blue iris are almost totally devoid of melanin, and as a result are transparent clinically. Lacking melanosomes, the cytoplasmic volume of the cells is also markedly smaller. This macro photo shows a light brown iris. Most of the iris pigment resides in the anterior border layer. A moderate amount of pigment is also present in the triangular wedge of ciliary body stroma and muscle at right. The dark brown iris of this African contains more pigment, which also is seen in the deeper stroma. The ciliary body stroma is deeply pigmented. In the past, some authorities thought that there were more melanocytes in darker irides. Histologic studies suggest that all irides contain a similar number of melanocytes. In my thesis for the American Ophthalmological Society, I examined a series of different colored irises using electron microscopy. I found that the size and number of melanin granules increases as the iris becomes darker. The largest melanin granules were found in a case of oculodermal melanocytosis, or the nevus of Oda. The round granules of neuroepithelial melanin in the iris pigment epithelium are always largest. This chart shows increasing melanin granule size as a function of iris color. The intersurface of a blue and dark brown iris are compared at high magnification in this pair of false colorized scanning electron micrographs. Lacking melanin, the processes of the melanocytes in the blue iris are slender, delicate, and dendritiform. In contrast, the melanocytes of the brown iris are swollen with pigment. Blue irides appear blue due to the Tyndall effect. Longer, less energetic wavelengths of light are absorbed or dispersed by the iris stroma. Only the shorter blue wavelengths of light have the energy to return to the observer's eye. In summary, a relatively similar number of melanocytes are found in all irides, irrespective of their color. Darker irides have more melanin granules, larger melanin granules, and hence larger melanocytes.